right, I wanted to put this in my pocket. I want to begin by thanking the organisers of the meeting, Nick and others. I've enjoyed it very much, and it's great to be here. I'm going to talk today on less than is in the abstract. And I guess first thing I wanted to begin with was the modelling of electrical activation is based on solutions of the reaction diffusion equation in an assumed continuum. What I'd like to do today is to persuade you that in actual fact this is not a continuum that we're dealing with, that in fact the heart exhibits discontinuous behaviour at around the 100, 200 micron level, which is at the level of scale which we're typically doing the work with modelling. I think that this is important in the normal heart, and I think it's even more important in structural heart disease. So I'm going to deal with normal heart and structural heart disease as we go through. Begin with work here, which is now old. Darren Hook's, in fact, tissue-based model of ventricular myocardium, published in 2002. He worked with a hand-segmented tissue block in which cleavage planes between layers of cells have been segmented out. And from model studies which he did with this, he made a series of predictions. They were that in fact that early activation from a point stimulus within myocardium was not continuous, it occurred in a discontinuous fashion. The laminar organization of myocytes, in fact, according to the modeling, determined three specific directions of activation. Most rapid along the fiber direction, which runs here, changes through the wall and runs there. Next most rapid within layers, and least rapid across layers, simply because these layers were presumed to be non-conducting, non-current flow boundaries within the tissue. Okay, the question I guess that I wanted to go forward is to look at is to what extent has the second of those two predictions in fact been borne out by experimental work, and a couple of studies which have been done recently, one led by Darren, where we've looked at the injection of passive current into the anterior free wall of the pig using a quasi three-dimensional array of dense electrodes. So these are plunge electrodes with 13 electrodes, plunge needles with 13 electrodes down them, current delivered from the central needle, the potentials generated by that passive current injection, passive current flow mapped out on the electrodes. For the entire volume contained within these electrodes, then the tissue microstructure defined, the fiber orientation, so we're looking at a plane here which is parallel to the epicardial surface, fiber orientation, but also the laminar orientation defined and characterized. And then the next phase of this work was to match experimental data, with modeled solutions, the Poisson equation in a tissue-based model, and to adapt the conductivities in fiber, laminar and laminar normal directions to best match these two. And in fact what was found here was very clear separation between the bulk conductivities in these three directions in a 4 to 2 to 1 basis. So microstructure defines three bulk conductivities, and that's been found for passive current flow. Very similar, though different experiments done by Brian Caldwell, coming out in Circulation, Arrhythmia and Electrophysiology shortly. And the way in which this experiment is done is pretty clear from the diagrams that you have here. A dense 3D array of electrodes, plunge electrodes in this case, pig heart again, the electrical activation mapped out in 3 dimensions from those electrodes, but in addition, again, the microstructure very carefully reconstructed in 3D across the total volume in which those electrodes are. And here we see isochronal maps rendered on the microstructure, on the microstructural representation, and a little bit more clear when we go to the next slide. Same slide, this is a mid-wall stimulus site, stimulating activation from a series of points through the wall of the heart. This is looking at the plane of the fibers, mid-wall parallel to the epicardial surface. This plane here is transverse to the fiber direction. And the guts of what we're seeing here in terms of this diagram is that the fastest direction of activation is, as we'd expect, in the direction of the fibers, and this is shown here, matching fiber direction with the maximum direction of activation. But if we look in the plane transverse to the fiber direction, we see in fact that the spread of electrical activation here is very non-uniform. This is for all experiments showing a spread in this direction, transverse, and that coincides with the laminar direction. And so again, experimental data, 3D data, demonstrating separation of the principal velocities, fiber, layer, and normal to the layer, again in a roughly 4 to 2 to 1 relationship. Whoops, wasn't what I meant to do. I think I'll move through the next slide, which is really just looking at the evidence for discontinuity. I think that's there, and just move on to the next work, which is now asking what do we see if we look at the border zone of an infarct. And here, work by Sally Rutherford, completing a PhD. This is looking at rat hearts with 14-day anterior infarcts, perfusion stained with picrocereus red, and these are then imaged transmural section in the border zone. So these infarcts are typically full thickness transmural infarcts, but we're looking in the border zone where there is a mix of infarction and viable tissue, imaged using the system which Ian showed you here. And this is just a series of 3 shots, just to orient us. The white there, probably not quite as clear as I'd hoped it would be, the white there simply showing the collagen, and those 3 shots as we come through bring us through to here. And so this is the epicardial surface, this is peri-infarct zone, infarct border zone, full thickness infarct to the left here, and some of the endocardium here shown up on that surface. 3 things that I really wanted to pick up. Firstly, the connective tissue from the infarct is driving down between the laminae, so we can actually see the layers in this particular view quite clearly, driving down and separating those laminae, and there's much less coupling between them. And we're still seeing in the infarct border zone 2 areas of preserved cells. In this case, an area of a ring of preserved cells around this vessel, and other preserved cells running up the endocardial surface on the side here. Okay, now what's been done with this has been to segment the tissue, so tissue segmentation, done at 2 micron level, and the connectivity between individual cells has been mapped out at this level using a connectivity mapping algorithm developed by Mark True. The process there enables us to look at the connectivity between cells, develop a connectivity map. Blowing that up a little bit more in the next shot, that connectivity map again showing the cell-to-cell connectivity, and using Greg Sand's structured tensor technique, the fiber orientations have been superimposed on that, giving us a model which in fact full structural model in which we can actually specify connectivity, and we've solved this as a monodomain model using the assumption that there's normal connectivity in the direction of the cells. So we've introduced anisotropy, cellular anisotropy, standard connectivity, and having done that, we've then in fact mapped out the activation times starting from a variety of sites around this as we go. And activation here from the epicardial surface, and the first thing to notice is that activation from the epicardial surface in this case is producing very, very slow propagation through the system, 91 milliseconds to propagate from here, activation ending up back on the endocardial surface close to this part of the peri-border or the infarct border zone there. The process of activation is slow because the pathway is tortuous, activation coming through here, down into the bottom, projecting up into the tissue, infiltrating back into the tissue via these long pathways. So we've got blocks here coming through again to here and then back down. So a tortuous process. that tortuous process is producing some of the slowing, but there's massive changes through this in the velocity. So local velocity profiles are changing, reflecting differences in tissue loading as we go through this tortuous process, reflecting also the fact that there are blind ends in the system which are in fact causing slowing. Okay, so we find that in fact that's true with normal propagation, discontinuous process as we've indicated, and we get different results if we stimulate from different parts of this particular matrix. So if we activate from the endocardial surface, the results are slightly different, and we can induce unidirectional block depending on where we activate and the loading conditions of that activation. Okay, I'm going to go through now and look at another related study, just asking what happens when you activate from a distance. What happens when you activate from a distance into a structure such as this, where there are lots and lots of discontinuities, lots of fibrosis. This is not quite specifically the same problem, although we hope to do this for the full infarct zone which you've seen there. So this is a simple representation, work by Zora Engelman. What we have here is a two-dimensional array of discontinuities, and this is a very simple model. We assume isotropy. We've assumed, we've used a Lua-Ridi-1 modified model in this case. We've changed the time constants to alter the time constant for slow sodium inactivation. And what you can see in terms of this experiment, a numerical experiment, is represented down here. We're comparing control with the situation where we have discontinuities, so a region of discontinuity activating from the top. We're looking in this case at two different states of activation, one with a long coupling interval and the other one with a short coupling interval, so slow heart rate, faster heart rate as we come down, and we're comparing for control and structure, structural heterogeneity. We're comparing activation time, repolarization time, and APD. And I guess the key thing to note is there's not too much difference between the two in this case, the longer coupling interval. For the short coupling interval, though, there's a clear impact of the structure, as you might expect, on activation. But even more striking, the structure is producing massive gradients of action potential duration, so huge gradients, spatial gradients of action potential duration as a result. Okay, now if we then come down and look at, whoops, not that, sorry. If we then come down and look at the end of a sequence where we've in fact been progressively reducing the coupling interval down, so reducing heart rate sequentially, we finish with a series of beats at a coupling interval 105, and we're looking at beats 11, 12, and 13 in the heterogeneous structure, looking at activation time and APD. And I guess really two things I want to say about this. The first is that as we move through the sequence, there is enormous electrical instability in this particular data set. We've got alternans occurring. The process of alternans is associated with massive changes of conduction velocity and activation times as a result, and the gradients of APD here varying from beat to beat in a substantial way, and that's reflected by the fact as we go through 1, 2, 3, we get block within the structure, which you're seeing here. There's block occurring in other places, but in this particular case, in the third beat, the block is occurring over quite a wide range of the structure, slow propagation occurring to the center, and that causes re-entry. The final slide I wanted to show was what that looks like if we actually plot a restitution relationship for that data set. So here we're looking at action potential duration versus diastolic interval. The open circles are control, that's no structure. The crosses represent structural heterogeneity, and we've got two cases. One's a steep restitution relationship, the other's a very flat restitution relationship. And what we're seeing here is that despite the fact that the cells are the same, despite the fact that the only thing here is structural heterogeneity, we're seeing changes in measured restitution, changes in the relationship between APD and DI. So here with a steep restitution, both control and heterogeneous structure, we're seeing instability, we're seeing alternans, but more of it and steeper for the heterogeneous structure. And for the lower case, where we've got, in fact, flat restitution, we don't see alternans in the control case, but we do see it, and we do see instability in the situation where we have a heterogeneous structure. So what I'm saying here is, I guess... Between A and B? Sorry? The difference between A and B? The difference between A and B is just simply that the control, I guess the cell properties are changed to give you a flatter control restitution relationship. So I'll finish there, and I guess I would just like to move on to the very end, having missed out some, and acknowledge the people who've been involved in the work and the sources of funding. Thank you very much.
Thank you very much. We're a little bit over time, so let us keep very short question.
Yeah, great. I really love that stuff. I have the following question. So, in the in-fight border zone, actually, due to the inflammatory process, fibroblasts infiltrate the area.
Right.
And so, in this study, you're assuming that anything that is not myocyte is not excitable, right?
Absolutely. It's
Yeah.
a very
However,
simple model.
Peter Cole has shown that specifically in the peri-infarct zone, myocytes form junctions with fibroblasts. So if you are to, instead of represent these regions as insulators, represent them as fibroblasts who do not have active properties but still represent a load to the current, wouldn't that smooth out some of these discontinuities?
It might well do that, and in fact I think there's two stages to that answer. One, I mean, obviously this is a very simple model where we're saying let's just take the simplest possible structural model and see what it does. And then that model is saying nothing has changed except that there's less continuity, less connectivity. Obviously we can add in the complexity and ask what, for instance, connectivity there fibroblasts might be doing. And the other thing is that it's absolutely essential, and this is much more difficult, it's absolutely essential to compare these kinds of detailed responses with experimental data. And we are doing that, both high resolution mapping in the sheep in-fight border zone, which I think is going to be harder to get down to the resolution we're talking about, but then also optical mapping in the rat. So, yep, so I guess that both in that model and the subsequent model on restitution, these are very reduced models, which are nonetheless producing results, which I think are worth following through.
And I would like to follow on the same question. So it would be probably also useful with some sections which are shaven off during connecting 43 and actually test this hypothesis if there is any coupling.
That is, in terms of the histology we're doing currently, the histology is more difficult, but it's something we certainly would like to do.
So, last question.
Yeah, I mean, your lovely talk. Wanted to address the issue of discontinuities between layers, rotating layers of myocardium.
Right.
In this case, what do you mean by discontinuity? It's clear that if you map this region, there are waves going across, there's a slowing down. So is this, when you talk about discontinuity, is it an extent of slowing down?
Yeah,
Whether you're going to call it discontinuity or slow conduct or slow propagation?
Yeah, I mean, the answer to that, I guess, I mean, the fact is, obviously, there must be, and there's high-level branching between adjacent layers of cells.
Right, and it's the same as transmural as the transverse.
Yep.
The two velocities are virtually identical.
Sorry?
Transverse and transmural are virtually identical
No,
velocities.
there's a 2-to-1 separation between the velocities.
What species?
In what we've just measured in the pig.
In the pig.
Yep, and that's...
We didn't see that in the guinea pig. It's the same as transverse.
Yeah, well, there's no... I mean, we've measured that both for... we've measured passive conductivities and active. So I think, and very carefully related that is high resolution to the structure. So 2 different studies, and it just, the same thing comes out of them two separate studies done by 2 separate groups of people. So it's a feature which is structural. It's not an active property inherently, so otherwise you wouldn't see it passively, and it's very clearly related to the structure in the pig. So that's the only animal that we've done that work in. That's consistent with what we believe, what we would expect in terms of the coupling between adjacent layers.
In terms of gap junction numbers?
Not in terms of, well, in terms of, I mean, there are space, in terms of laminar structure, the way in which cells are organized in layers, layers contain groups of cells, 4 to 5 cells thick, which actually have tight coupling. So within any layer, it's just like you would expect it, tight coupling, each cell connects to each other. Between layers, you then have branches which are higher order, and gaps which are on the order of maybe 200 to 300 microns in some cases. It depends where you look. So you've got relatively large areas where, in fact, there is no apparent cell-to-cell, no apparent side-to-side coupling. And you can see that histologically when you actually go through the tissue, you can actually find what those clefts are. And I guess what the experiment's showing is that the results map back to what you might expect from three-dimensional reconstruction of the histology.
All right, well, thank you very much again.